I'm going to talk about bridge that is a methodology that we implemented in our lab. Our lab is located in Trieste, Italy, in the International Center for Genetic Engineering and Biotechnology. And we are working in Saccharomyces. And my talk will be about bridge translocation, and that's why there are many pictures about bridge, like this wonderful here. Okay, I will talk about translocations between heterologous and homologous chromosomes in it. And these are a few of the people of the students involved in chromosomal translocations between heterologous chromosomes. And these are the related publications about chromosomal translocations. The system is quite simple. I don't know if you are familiar with Saccharomyces cerevisiae. It's an eukaryotic cell with a diploid genome. It can be also haploid, but we apply the system for the diploid cell. And these are two chromosomes. Chromosome 8 here that you can see, and chromosome 15. And this is XXX, which is bridging physically together the two chromosomes. These are full of CR. These are metabolic things, so nothing to do with recombination. And physically, we can bridge together these two chromosomes and produce a translocated chromosome that can be easily selected on the ovary because we give the selection portion of the These chromosomes can be easily selected and follow along also the life of the cell. And here is another example. The cassette is easy to prepare because you can prepare it by PCR, and then transforming yeast cells, you can select for the transform and check the translocants. So the system is quite efficient, but it depends on the length of this cassette, so on the homologies, of the length of the homologies. Increasing the length of the homologies, you can have the increase of the efficiency of the system. When you have 14 nucleotides, for instance, of homology, the efficiency of the system is roughly 2%, which means that you have two translocated cells out of many transformants that you can find on a plate, on a G218 plate. When this homology is extended up to 65 nucleotides, 70 nucleotides, you can reach 5-8% of the efficiency. It depends also on the recombinogenicity of the place, because in yeast genome, as in other genomes, also in mammalian cells, there are few places that are hotspots for recombination and others that are cold spots. So of course it depends also on the recombinogenicity of that place. Promoters, for instance, are easy targets, so we can say easier to be targeted than intragenic regions. And you can see this is a southern blot. You can easily check the translocated chromosomes. These bands that you can see here below are rearrangement of the translocated chromosome that can happen during the life of the cell. So it's easy to check the formation of the translocated chromosomes also by PCR. So using primers at the junction on the genome, you can easily verify if the cassette produces the translocation. And this is the bridge. Using this primer and this primer on the two different chromosomes, you can amplify the bridge between the two chromosomes, in this case chromosome 5 and chromosome 8. And this is the southern blot with which you can verify the formation. This is chromosome 8 that here was probed with kanamycin to verify it on the gel. This is the size of chromosome 8, and this is the size of your translocated chromosome, which is smaller with respect to chromosome 8, as you can see. So by southern blot, it's easy to visualize the formation of the translocation. So the technique is possible, but we demonstrate that the frequency depends on the recombinogenicity of the strain of the loci. Increasing the homology, the frequency of the translocation increases also, and if you go to 60 nucleotides, usually it's 5-10%. You can have a various panorama of events. You don't have only translocants. Only 2 or 5% of your transformants are real translocation events. All the others can be simply recombinants with other gross chromosomal rearrangements events. For instance, you can have ectopic integration. What does it mean? It means that the cassette jumped in somewhere else. It doesn't give you a translocation. The cell is resistant on the RNA, but you don't have a translocant. You can have simply the integration in one place. This is quite common, also because there are many hotspots of recombination, especially in the yeast genome. Then you can have aspecific translocations. So the cassette is integrated not in the place that you want. For instance, exploiting a microhomology. Let's say we have 60 nucleotides of homology, only 30 of these are integrating in another place. You have a translocant, but it's not the translocation that you want. Or you can have a combination with 2 micron. 2 micron is an endogenous plasmid of yeast. Yeast has the majority of the strains of yeast, the natural yeast, they have this plasmid inside. It's not a drawback or an advantage, this plasmid, to have this plasmid. This plasmid is named 2 micron because the size of the plasmid is 2 micrometers. And it stays in the cells, but it can recombine. It has a high efficiency of recombination. So if you're transforming yeast with DNA, exogenous DNA, it can recombine also with 2 micron. So you have to keep in mind when you're transforming yeast cells that many of your transformants probably will be transformants due to recombination with endogenous plasmid inside the cell. And that's what I said before, that strong constitutive promoter may correlate with the recombination integration because they are more prone to recombine. You can have also secondary rearrangements due to transposons that are present in yeast as in other organisms. And interesting thing, we also demonstrated that the same translocation event with exactly the same cassette, with the same homologies, may lead to different aneuploidies. So you can have that these translocants, when you compare them, they can have also different secondary rearrangements and different numbers of chromosomes. Because one single translocation event may lead to many different aspects, so many different we can say genomic rearrangements, and the translocants are never exactly identical each to the others. So they are different, always different among them. This was just to show you what a translocation can trigger. These are normal yeast cells that were labeled with DAPI so that you can visualize the nucleus inside the cells. And this is after the translocation event. And you can see the aberrant phenotype of these cells that are resembling the cancer cells. And these cells are aneuploidic cells with, of course, more chromosomes of the natural cells. And in this case, they have at least the chromosomes that we induced, the translocated chromosomes. And then they can have also secondary rearrangements and different secondary aneuploidies. And this is with DAPI, the staining of the nuclei of this cell. And you can see that many cells are without the nucleus, and you can have also the fragmentation of the nuclei in many other cells. So the event of the translocation is really important in the sense that it can bring to really aberrant phenotypes, so a complete transformation of the cell. One single translocation event that you induce is producing a complete genomic rearrangement, resulting also in a complete aberration of the phenotype. And this is one of the reasons for these aberrant phenotypes that around the breakpoint These are the two chromosomes. This is the cassette, this is the bridge. And you can see that around the break point, you can have an increase of the genes, of the expression, of the gene expression. These two violets, the stronger and less strong, let's say red and violet, these are two different translocans analyzed. And you can see that for both of them, you can have an increasing of the expression of the genes around the translocation breakpoint. And of course, this is translating in an aberrant phenotype, because you can see that when you change up to 5 times the expression of many genes, of course, you can have an aberration of the phenotype. Because these genes, of course, they code for proteins, so the stoichiometric, we can say, ratio of the proteins changes, and the interaction and interactome is completely changed, because with one translocation event you can have an alteration of many genes, and therefore many proteins inside the cell. Okay, this is Le Pont du Gard, which is a bridge in French, just to remember that it's bridge in this translocation. And we studied, these are still unpublished data, we studied also how the bridge-induced translocation correlates with the cell cycle. In yeast, it's very easy to study the cell cycle. So to stop the cells at a certain point of the cell cycle, and therefore it allows you to study a single event at a certain step of the cell cycle. And these were the two people working with me in this part of the project. This is the cell cycle of the yeast cell. We have G1, S, G2, and phase. So you can block the cell at one stage of the cell cycle. For instance, with hydroxyurea, you can block the cells in S phase during the synthesis. These are the cells blocked during the synthesis. So we repeated the bit, so we transformed with the cassette with the two homologies at this step. And what we found is that the translocation frequency increased up to 15%. Then we blocked the cells with nafcodazole at the end phase. When you have the nuclear division, you can see here that the nucleus is migrating from the mother to the daughter cell. And we repeated the experiment, the bit, and we found the translocation frequency dropped down to the 0%. So it means what? That usually synthesis promotes bridge-induced translocation event. So you can have, now we are testing also G0, G1 stage, and we found that we had few events of translocation. So therefore, the majority of translocation events with these cassettes are focused on S phase, a little bit in G1 and 0 in M phase. Then we wanted also to verify if it was possible to repeat the same translocation event, but not between two different chromosomes in a diploid cell, but between homologs. So for instance, in a diploid cell, we have two chromosome 2, two chromosome 3, two chromosome 8, and so on. And we wanted to bridge together the same chromosomes. And these are the people working on this part of the project. For instance, we had two chromosomes 8 that we labeled, of course, differently, and we repeated the experiments between the two homologs to see what? The efficiency, first of all, also compared with translocations between heterologous chromosomes, and then if the translocation was preferred versus the knockout, the deletion. What does it mean? These two chromosomes are identical, so this sequence is identical here and here. So this cassette is producing a translocation, or it's integrating on the same chromosome, producing a deletion in this case, because these homologies are exactly the same on both chromosomes. And we found that the translocation is still produced, but the efficiency is really low. I'm not entering details on this. It was just to show you that the mechanism of the translocation between homologs, that you have the invasion, and then when the holiday junction is formed, this is migrating, this DNA is missed, so you have the degradation of this part of the chromosome, and at the end, the resolution. So that at the end, you don't find here a non-reciprocal translocation, as we usually see between heterologous chromosomes, but a near reciprocal translocation. And at the end, since you are losing DNA, you have loss of heterozygosity. So this piece is not present anymore in the two homologs, in one of the two homologs. And also looking at the expression between homologous chromosomes, we don't see a lot of difference. If you remember before, I said you have an increase up to 5 times around the breakpoints during translocation between heterologous chromosomes. But between homologs, we don't see this alteration of expression. The only genes which are altered are genes related to the cell cycle or to multi-drug resistance. And therefore, there are a lot of differences between translocations between different chromosomes, so heterologous translocations or homologous translocations. And the main difference is that the frequency here is lower, and it's roughly 1% maximum between homologous. So it means that the cell doesn't like a translocation between homologous chromosomes at all. And then, if we compare now the mechanism between heterologous chromosomes, you have a non-reciprocal translocation. Between homologs, you have a near-reciprocal translocation with loss of heterozygosity. Then, around the breakpoints, between homologs, you don't have alteration of gene expression. Between heterologous chromosomes, you have a lot of alteration of gene expression. And some, we can say, cell cycle-related proteins are in any case affected. And in both types of translocations, they are always scrambled up. So they are really completely affected and deregulated. And therefore, of course, everything means that this bridge-induced translocation, the translocation event, is a destabilizing event for the cell. And it's regulated by some checkpoints and some genes. So we wanted to, of course, to look at which are the molecular players or the architects bringing to the translocation event. And this girl was an undergraduate student. She worked a lot with the molecular players, trying to understand which are the genes involved in the process. And in particular, we focused on RAD54, RDH54, and POL52. And the content I'm going to discuss a little bit now is now in this paper, which was accepted last week. So this is the mechanism. We wanted to see how does this bridge, so the translocation end up. In other words, after the first integration event, this is a model that we proposed, you have the degradation of this part, or its rearrangement in the genome, because we can find it's rearranged sometimes in the genome. And then you can have here, not an integration, but a copy. So that's why you have non-reciprocal translocation event. You don't have two breaks. You have one break, and then with a mechanism that is named BIR, break in this replication, you can have a copy of this part of the chromosome. It means that at the end, your cell will have this piece in 3 copies of the chromosome. This piece of the chromosome, this is chromosome 8 or 7, it will be in 3 copies in the cell. So to verify it between heterologous translocations with 2 chromosomes, we generated a series of mutants. These mutants are mutants of genes that are known to be involved in some different aspects of translocation, of chromosomal translocation events. All of these genes are involved in chromosomal translocations. This is the wild type that is named SUN1, because a student and former colleague of mine, Sanjeev, generated it, so it's named SUN1. And these are the mutants. These mutants are double mutants, double deletants, which means that, for instance, ELG1 delta, it means that the gene ELG1 is deleted in both copies on both chromosomes. So you don't have the 2 copies of ELG1 in the cell. Of course, all of these genes are not essential genes for the cell. You can see also here the phenotype already of the mutants. So missing these genes, the cells, the yeast cells, have particular phenotype. This in yellow that you can see here, this is a staining named fan staining. And with this staining you can see the dead cells. So these in yellows are all dead cells. Okay, I'm not entering details in this. This was just to show you that this is the wild type, and these are the mutants. And these bars with different colors are the different veins, which means integration only on one end, integration on the other end, translocation, or ectopic random integration of the cassette. And you can see the difference between the one type, just by a glance, and the other mutants. So you can have here, for instance, the gene SGS1, which is the gene, it's the homologue in the human cells of the Werner syndrome. Okay. And when this gene is missing, you can see that you have an incredibly big amount, enormous amount of ectopic integration events, which means that the cassette is jumping in the genome. And it's captured by the genome, and it remains there. Why? Because it's known, for instance, that when SGS1 is missing, the genome is very flexible, and it's very prone to capture all the exogenous DNA that is inserted into the cell, for instance. But you don't have, in this case, translocation events, which should be violated. So, for instance, in this case, when this gene is deleted, you don't have translocations. For RAD54, you can see something very, very strange and particular. That is the fact that you don't have any event, which means that when this gene is deleted, you don't see integration, translocation, integration in somewhere else, recombination. This gene is very important, and it's a gene of recombination, and when it's missing, and we will see also when it's overexpressed, the big system completely changes. Okay, here it was just to show you a few of the phenotypes in the translocated cells. And you can see these are translocants in the mutants. You can see these that are alpha formation, many dead cells, strange sizes of the cells, and therefore the mutants, two nuclei in the same cell, for instance. These are few aberrations, the translocation is. Okay, when RAD54 is missing, you don't have translocations. When it's overexpressed, you have many cross chromosomal rearrangements, which means that you can have, you can see here, this is a sudden blot, this is a shaft, which is a separation of the chromosomes from the gel, and this is a sudden. And you can see that many veins are appearing. Why? Because when this gene is overexpressed, you can have many recombination events. This is known also in cancer cells. In prostatic cancer, for instance, when they found that this gene is highly expressed, RAD54. And these prostatic cells are prone to be strongly aneuploidy, to have aneuploidy and to have many rearrangements. And now we demonstrated why, because you can have recombination between long terminal repeats that are interspersed in the yeast genome, and you can find them also in mammalian cells, these repeated regions. And when RAD54 is overexpressed, you can have the recombination between these repeats. So after the primary translocation event, you can have secondary rearrangements triggered by the loss of this gene or by the overexpression of this gene. In this case, when this gene is overexpressed, you can have many rearrangements. RDH54, we demonstrated that it's regulating the orientation of the resolution of the allele junction. In other words, when you have the invasion, then this is the wild type, this is the mutant without RDH54. So in this case, you can have the allele junction migrating in one direction. When this gene is missing, the allele junction is migrating in the other direction. And you can easily guess that the result is completely different, because in this case it's this strand that is degraded and not the other one. So the result will be completely different at the resolution point of the origination. And then we analyzed also Pol32, and we demonstrated that this gene is not correlated with the formation of translocations, at least in yeast cells, and probably also in mammalian cells, because all these genes have a normal in mammalian cells. So the translocal that you can see here is normally formed. So in all of this, in all of the mutants in which Pol32 is missing, the translocation is possible. The number of the translocated chromosome varies a lot. You can have 2, 3, 1 copy of the translocal. And this is depending on Pol32 absence. And I want to conclude with bridges, because this talks about bridges. So bridges from my country, but also from other countries. This is a famous bridge called the Street in Venice. This is the oldest stone-built bridge that is Ponte Vecchio in Florence. The oldest bridge that is stone-made. The longest suspension bridge, the Golden Gate in San Francisco. The symbol of London, the Tower Bridge, one of the most famous, of course. The bridge connecting Sweden and Denmark, the Øresund Bridge. Just to demonstrate what also men are building. The widest long-span bridge in Sydney, Australia. In San Diego, United States, is one of the tallest bridges of the world, the Coronado Bridge. It's connected to another bridge. In China, the longest bridge ever built, an ocean crossing bridge. Hangzhou Bay Bridge, I don't know what the pronunciation is this one. Then a bridge made of water over water. This is very nice. This is in Germany. And then a spiral bridge to minimize the space that was built in Shanghai, China. This is in France, and it's the largest cable-stayed particular bridge in the middle of France. It's really impressive. And then the oldest bridge, original bridge perfectly conserved, was built, of course, before Christ, and it's Ponte Fabricio in Rome, which is in the center of Rome, in Isola Tiberina, that is an island, inside of the city. And then I finish with a bridge that is not famous at all, like this one, but it's of my town. That is the Ponte Rosso, which means the red bridge. It's not red, so I don't know, don't ask me why it's named the red bridge. But it's in the channel inside my town, that is located, we can say, near Venice, in northeastern Venice. And this is the Ponte Rosso. So thank you very much for your attention and for the invitation.
Just a point, I mean, you described the fact that the frequency of conjugation is rapidly increased when septa or physically essential to the urea, which is blocking it.
Yes.
Basically, but I'm not sure if I got the mechanism of the explanation for such a big increase.
Yes.
Is it strictly linked to the fact that that's carried the drug, or is it the recovery after the drug that will
No, it
lead?
links the fact that, if you remember, when I explained the mechanism.
Basically, I do not remember.
Okay, I can show you.
Thank you.
Here. Yes. It depends on the fact that the majority of these translocation events are due to a first integration event, and okay, this is dependent on drug 54 and drug 56, we can say epistemic of genes. And then the second step is due to BIR, so the so-called replication, breaking this replication. And therefore, usually, when you are in synthesis, this event is strongly promoted. And therefore, when you block the cells in a space, especially if you are very near the telomere, this event is promoted. And therefore, you have an increasing of the event of the efficiency.
Thank you.
Thank you for your presentation. Is there any clinical application on this project? On this subject that you did or you are doing now?
Well, we want to try also to expand this project to mammalian cells. The main problem that we have is that differently from Saccharomyces cerevisiae, in mammalian cells, the homologous recombination system is very, very low. And this system is based especially on the spontaneous homologous recombination system of Saccharomyces cerevisiae. So my guess is that if the efficiency is so low here in yeast, it would be very extremely low in mammalian cells, where the homologous recombination system is not working well. Because usually mammalian cells prefer non-homologous adjoining or single-strand annealing to repair the breaks, and not homologous recombination. But we want to apply this system in mammalian cells, probably overexpressing recombination genes like RAD54 on RAD52. In this case, the efficiency of homologous recombination rise in mammalian cells, and you can apply this system also in mammals. But usually, just keep in mind that in mammalian cells, homologous recombination system is extremely inefficient. This is why it's so difficult to apply the system as it is to mammalian cells.
Thank you for your presentation. I have two questions. If you understand that collagen which regulates voltage junction can be built bridge?
I don't understand. I understand.
If we understand that collagen which regulates voltage junction can be built bridge?
The RDH54, you mean? The gene regulating the holiday junction is RDH54.
Yeah.
This gene doesn't have any effect on the translocation itself. It just moves the holiday junction, but it does not increase the efficiency of the translocation. But it's involved in the stability of the translocant, the chromosome that is formed. When this gene is missing, the translocated chromosome is lost in the next generation, so in the lifespan of the cell, with higher frequency. But in the frequency of the event, of the generation of the translocated chromosome, it doesn't have any event, any effect.
Which chromosome translocations most important characteristics of cancer?
Okay. To answer these questions, there are many translocations that are related to cancer cells, and the majority of leukemias and lymphomas are due to translocations. And actually, we have a project in the lab to reproduce perfectly a translocation that causes a chronic leukemia in humans, and between the NUP and the top NUP gene and the topoisomerase gene,
in humans, the fusion of these two genes is producing a chronic leukemia. So we decided to do the same between the homologs in Saccharomyces cerevisiae, to fuse these genes and to see the effect in yeast and to check to find for the regulator of the genes. But we are still building the fusion because it's not easy to reproduce. But we are working on that. It's a project just started 2 months ago. So usually all the leukemias are typical due to translocation in mammals, and the majority of them are reciprocal translocation. Few of them are also non-reciprocal, as the one of the reproduced translocation.